already, so I'll try to be brief. And I'm going to be talking a little bit around interface and the relationships around between aptamers and nanostructures. And during the COVID years, this is a lot of the sort of direction of our lab at the University of Hong Kong in terms of some of the things we've been working on a little bit more and moving a little bit more towards. So our lab at HKU, and we have a couple of labs there. We've got one at the university, we've got another one at the Science Park, at the Advanced Biomedical Instrumentation Centre. We're really across 4 areas. What I'm going to be talking about today is in aptamers, diagnostics, therapeutics, and particularly aptamers and nanostructures. I'll touch a little bit on the abiotic chemistries. And this area, we're doing quite a lot in terms of droplet-based selections. If anyone would like to chat about that, I'd be happy to in a coffee break, but I won't be talking about that today. And in particular, thinking about aptamers and nanostructures and how they can help each other. And actually, when I was preparing these slides, I was looking back from some slides when I last presented here 4 or 5 years ago. And I had these 2 questions around what limits nucleic acid function, then what's special about nucleic acids. What's and you know, many of the talks from the program today we're going to be looking at the amazing things happening in the chemistry, going beyond 4 varieties of base and so on. But also I think an important thing is thinking about how aptamers displayed, and of course their stability, but nanostructures can help with aptamer display. And then what's special about nucleic acids and why we're using them instead of antibodies for molecular recognition. And one area which has really changed radically in the last 10, 20 years is around DNA nanotechnology. There's been really exciting developments there. And of course, the other reasons, PCR, of course, has allowed CLEX and in vitro evolution and the increased conformational states per unit compared to proteins, allowing all sorts of different structures of single-stranded nucleic acids. I'm going to focus on this one around DNA nanostructures. And in the DNA nanostructure field, thinking about sorts of things that one can make. Now, one big area, of course, has been the DNA origami. In terms of translational application of DNA origami, that can be a little bit limited because when you're making DNA origami, you need 200 oligo staple strands to be building up all the structures. So, of course, the Rotherman group, the classic paper from this must be over a decade ago now, of them being able to make any structure they would like. That extended up into 3 dimensions. One can build all sorts of things out of origami. So it's been amazing, but it has this limitation, particularly when you're thinking about applications of origami in translation, in drug delivery, and so on. Another slide where there's been a lot of investigations around small polygon type structures, particularly the tetrahedron, benefit there, small number of oligons. But not very, it's difficult to think of building nanomachines out of these sorts of things. And you know, sometimes these can be limited in what you might be able to deliver or use them for. So a question we asked in the lab was, can we use a small number of oligo strands but also create large complex structures? And this is a question which we had been looking at. And then I was visiting actually a museum in Venice, and I saw this structure of the something called a mazzucchio, and it's something which Da Vinci had written on back in the 15th century on the use of mazzucchio, and it was represented a lot of artwork at that time. And I sent this to one of my colleagues in the lab, Dr. Simon Sue, a picture of it, that's the actual photo, which I took in the museum, and said, can we build this out of DNA? And then he managed to do so in terms of a particular structure here with a hexagon, this first generation hexagonal. This structure was quite flexible, so we weren't able to get very good TEM in this first generation. And the sizes of it were as we expected. And then we moved out to another second generation of these, which is a little bit more rigid in terms of these mazzucchio structures. Octagonal, each subunit of these going round. And one can see this in TEM, one can see this in AFM, And at the moment, we're looking at this by cryo-EM also, and they're the expected size, both by TEM and AFM, for building up these maxillary structures. So these carry the advantages of being large structures like origami, but they're only made from 12 oligons in this case to build them up. And then that opens up all sorts of possibilities, particularly as we think about how to integrate with aptamers to functionalize. What we looked at in one of the applications of these maxillary structures in terms of targeting, in particular, nasopharyngeal carcinoma. NPC is a major problem in southern China. In China, it's one of the major cancers. And one of my colleagues at the Chinese University of Hong Kong, a different university in Hong Kong, had developed an aptamer called APT-20119, which she had, her lab had selected by cell select against NPC tumor cells. And we had attached the 2001-19 aptamer onto both the tetrahedron and onto the maxillary, and then comparing the effects of the tetrahedron and the maxillary in terms of delivery of nanostructures. So we had looked at delivery here, just using a fluorescent label of the nanostructure. You see the comparison of the tetrahedron on the left with the maxillary on the right. At the top, without the aptamer. At the bottom, with the aptamer, you see that the aptamer does help the delivery of the DNA structure. And the maxillary seems to be a little bit better than the tetrahedron in terms of delivery of that nanostructure. We looked at the mechanisms, and the mechanism, first looking at this data here, just notice on the y-axis that the number at the bottom here is much bigger than the numbers at the top. So it's much more effective with the aptamers relative to just the naked DNA nanostructure for the delivery. When there is no aptamer, then delivery tends to be primarily through macropinocytosis. But then when it switches, you've got an aptamer against one of those receptors on the cell, on the NPC cells, and you see this switch to dynamo-independent, clathrin-dependent endocytosis with those particular aptamers. We then went on to doing this with xenograft animal models, NPC tumors in mice, and you see that the maxillary and the maxillary labeled with those NPC aptamers is more effective than the tetrahedron in delivery to those tumors, both for the whole mouse and then for the tumors alone, taken from those mice. And it also appeared that that large DNA nanostructure more resistant to kidney filtration relative to the tetrahedron. So for that story, that's where we are in terms of that application of the NPC, that we were able to put the aptamers on these DNA maxillary, use them for a particular delivery, and this was specific to NPC tumor cells. We have some data around the mechanism of that, and so of course that could be very useful for delivery of drugs, delivery of doxorubicin, or it could be used in imaging, and we could be using that large maxillary structure for all sorts of imaging nanoparticles as a way to carry them there for that. The other aspect of this sort of rotary structure, which we're really excited about, and this is our sort of most exciting project right now, actually. Simon was looking through another, after we found out that there's the Da Vinci sort of, looking through one of the sort of Da Vinci books, he saw one of these structures. There's another image from Da Vinci around a mechanism, rotary mechanisms. And that's another possibility of these sorts of nanostructures. So if you've got some sort of motor on a rotor, then what you need is some sort of fixed base also. Yeah, and there's quite a lot of research in the nanostructure field around these sorts of triangular origami. So people in the nanostructure field, you see this quite often. But then what we've done is we've put the maxillary on the top, and you can see this by the TEM. So you see the triangle at the base, and then this rotary maxillary which is sitting above it. Okay, And then this starts to open up lots of other ideas around molecular machine applications of this, and perhaps using aptamers or aptamerzymes for that. So as a first proof of principle, we've demonstrated this as a particular clock. So the clock here has two fluorescence tags. So we've got one red. So the red bit is a Psi5 on the maxillary. Okay. Now underneath on the origami sort of base, it's got a Psi3 in one low corner, and it's got a quencher in the other. A quencher in the other one. The black one is the quencher, green is Psi3, red is Psi5. Okay. So in this position at 12 o'clock, you should get fluorescence from both the Psi3 and the Psi5. They're not quenched, and that's indeed what you see when you're measuring the fluorescence. Okay. Then if you add oligos to sort of move around and then through competitive process, move this maxillary around rotationally. So now the Psi5 is above the quencher, should get quenched. It gets reduced at the 4 o'clock position. But then you still have the fluorescence here from the Psi3, which is on the other corner. Then it moves around to 8 o'clock. Then you get the fret that got Psi3 and Psi5 next together. And then you've got the original fluorescence of the Psi5. And the Psi3 itself will be reduced because it's all gone through the fret. Okay. And then back to 12 o'clock. And you can push each of these transitions by adding particular oligos, which are complementary to those ones interfacing between the origami and the maxillary above. So we sort of have that system going, and this could be really useful in terms of thinking about rotary mechanisms, because what we would like to do is be thinking about aptazymes or DNAzymes being positioned at 3 places, because if you have some sort of rotary mechanism based on this sort of structure, then that could be useful for quite a few different applications. So it's another example of where aptamers can help nanostructure function. So one is the classic in terms of cell delivery, another is in terms of these rotary mechanisms. And then I think if you go on to using chemistry, it's a long way beyond the typical AJCT. So for example, the collaboration we had with Marcel for using cubanes within aptamers, which was really stimulated by this conference. I think this photo maybe 6 or 7 years ago. And then Marcel's amazing nucleotide chemistry to make these cubane type analogs. Then we solved the crystal structure of the cubane aptamer together, this with the malaria biomarker, PV-LDH. These sorts of things, and then using that sort of abiotic chemistry on that sort of motor could be really quite exciting. So these are sort of directions we're thinking about. Now, the other side of this talk is, because we're talking about that symbiosis of how nanostructures might be able to help aptamers. And then I was thinking back to something we published a few years ago, here around surfaces. Then you've got an aptamer on it. So it could be for electrochemical ones, it could be for, this one is a colorimetric assay. But often it would be better if the aptamer is away from the surface. And DNA nanostructures can do that very uniform at the molecular level, at the nanostructure level. So for example, if you put the aptamer on top of a tetrahedron, then it increases the efficacy of this assay. This is a particular assay, which is colorimetric, of a protein binding on top of this aptamer. And in red is for the tetrahedron, and black is just the aptamer. So nanostructures can help aptamer function as well. And this is more recent work that we published just last year around tiling on a surface, here for DNAzymes. But it's just the thought that if you've got some way of displaying aptamers very uniformly across a surface, every 100 nanometers or something, then it can lead to much better activity. In this case, DNAzyme activity. It's the DNAzyme which we DNAzyme was higher activity. See that tiling by tiling. But this is the way that nanostructures can help aptamer function. It's not always nanostructure. So this is, I think, my final slide, that we have the symbiotic relationship with atoms and nanostructures that can help both. Something to think about. So just my final slide here. I'd like to thank Anna Tang for, in particular, NEC collaboration, Chinese University of Hong Kong, Marcel Hollenstein, fantastic collaborator in terms of industries. Dr. Simon Hsu and Dr. Lin Wang really pushed forward all of this nanostructure work in my group, many other group members, funding for this particular JRF. And just as a last message, we are recruiting a postdoc in Hong Kong right now. The deadline's in a couple of days, but an exciting place to work, and there's a lot happening there. If anyone is thinking quite translationally around spin-out companies in Hong Kong, Greater Bay Area, China, it's a good place to be. Okay, so thank you very much.